Hello, everyone, and welcome to today's webinar, Insights, Ultraviolet Germicidal Irradiation for High-Level Disinfection of Ultrasound Probes. I'm your host today, and my name is Prof, the Vice President of Marketing and Communications for Germitech, and we are sponsoring the webinar today. You have joined the session through the on-air live streaming webinar portal. Due to time constraints of our presenters, we won't have a Q&A session at the end of the webinar. However, we will provide you with an email link for submitting your questions to our presenters, which will be included in the follow-up email containing today's webinar recording. Here is an overview of today's webinar. High-level disinfection has traditionally been performed using chemical germicides like glutaraldehyde, parasitic acid, hydrogen peroxide, or OPA, all of which have drawbacks. More recently, automated systems based on ultraviolet UVC irradiation have gained approval in various countries for performing high-level disinfection on non-lumen devices. These devices include ENT scopes, T or toe probes, as well as vaginal, rectal, and surface ultrasound probes. In this webinar, we review one UVC device, Germitech's Kronos system. Kronos high-level disinfection microbial efficacy is compliant with various standards, including EN 14885 and the new British standard BS 8628 2022. And the webinar provides insights in how this is achieved. We will further explore this system in light of recent published clinical studies and conclude with a Mythbuster session dispelling misconceptions related to UVC high-level disinfection. Today's learning objectives are aimed to provide participants with a comprehensive understanding of UVC-based high-level disinfection, its benefits, regulatory compliance, clinical evidence, and its role in enhancing infection prevention strategies within the healthcare setting. I would like to now introduce you to our two presenters. Our first speaker, Dr. Lionel Penu, a PhD in Microbiology, Scientific and Innovation Director, European Biotech GERMAD. Dr. Lionel Penu was the original founder and current Scientific and Innovative Director of European Biotech GERMAD, a company dedicated to the evaluation of medical devices, environmental sampling in health settings, and training in hospital hygiene. His expertise in the field of endoscope and medical device reprocessing is recognized worldwide. Lionel Penu participates in numerous ISO commissions in the field. Our second speaker, Professor David J. Weber, is a board certified in internal medicine, infectious disease, critical care medicine, preventive medicine. His research career has focused on healthcare-associated infections, antibiotic stewardship, new and emerging diseases, and vaccine implementation. He has published over 550 peer-reviewed scientific papers. I will now hand you over to our first speaker, Dr. Lionel Penu.
Good morning, ladies and gentlemen. My name is Lionel Pinu. I'm the Scientific Director of Eurofins Hospital Hygiene Business Line. I'm very happy to host this webinar on UVC disinfection process and share with you the results obtained on the Kronos unit. During this presentation, I will first very briefly remember you how UVC works, and then I will describe the test done to demonstrate the antimicrobial efficacy of the Kronos unit. And I will also explain you how we can demonstrate that this process reached the same efficacy level that what is required for a chemical disinfectant intended to be used on a medical device. Then at the end of the presentation, I will also discuss the interest and efficacy of such process on specific microorganisms like HPV and bacterial spores. So first, regarding the mode of action of UVC. It is well known that microbial inactivation process by UVC radiation is mainly based on the occurrence of photochemical reactions caused by UV light on genetic materials of microorganisms like DNA or RNA. In fact, to be precise, the adenine-thymine bond of RNA or DNA is broken by UV light, affecting the transcription of this RNA or DNA, resulting in the inactivation of the microorganism. But there is also some other evidences showing that photo can also interact with cell envelope components and promote the oxidation of unsaturated fatty acid, leading to the inactivation of microorganisms. So now we know how UVC inactivates microorganisms, but how can we demonstrate that this process can achieve high level of disinfection? In fact, it's quite simple. We just have to follow what is described in the EN 14885 standard. This document specifies all tests to be done for a chemical disinfectant to demonstrate or to support a specific claim, like for example, bactericidal activity or virucidal activity of a disinfectant intended to be used on a surfaces or on a device or on a medical device. For this purpose, Germitech works with scientific centers in France, like Eurofins Biotech Germain, but also in the United Kingdom, in Germany, and in the United States with the Penn State College of Medicine to independently demonstrate the efficiency of the Kronos unit. So what are the tests performed to demonstrate that such a UVC disinfection process works? Mainly two types of tests have been performed. The first one, called carrier test, and the other one on real use condition called clinical use test. During this presentation, I will focus on carrier test, and David Weber will develop the clinical use test. Regarding EN test, it is important to know that according to EN 14885, there are 4 types of tests. Phase 1 tests, which are basic suspension tests, not used for efficacy claim, just used to demonstrate the activity of active substances. Then we have phase 2 step 1 tests, which are quantitative suspension tests. But these tests are suspension tests, and because they are suspension tests, they cannot be used for UVC disinfection process, and they are only applicable to chemical disinfectants. Phase 2 step 2 tests are quantitative carrier tests, and these tests can be used on UVC disinfection process. We will see later on how it's possible. And then we have phase 3 test called field test or clinical use test. So coming back to phase 2 step 2 tests, of course, because they are carrier tests, they can be adapted or modified to be used for UVC disinfection process. Mainly what we change the surface to be tested. We use silicone instead of glass or stainless steel because silicone is more representative of the device surfaces to be disinfected. The tests were performed on clean condition, that means with a low amount of interfering substance at the minimum UV dose corresponding to the minimum contact time of the disinfectant. Tests were performed on all required microorganisms, plus some microorganisms of clinical interest, and the efficacy requirements were the same that those required for the chemical disinfectant. In addition to what is required in the EN standard, we have also performed some other tests called simulated use tests, which are tests done on artificially contaminated props using the most resistant strains in the presence of interfering substances. For all carrier tests, to claim high-level disinfection, in fact, you have to demonstrate that you are able to achieve a certain log reduction based on the EN or the standard you use. And this log reduction may change from one microorganism to the other. For example, according to the EN regulation, you have to demonstrate a 5 log reduction in bacteria, whereas for viruses, you just have to demonstrate a 4 log reduction. And as you can see on the right part of this slide, all tests done with the UVC disinfection process were compliant with the standard requirement. And we can clearly say that the Kronos unit reached the same efficacy level that what is required for a chemical disinfectant intended to be used for the disinfection of medical devices. You have on this slide the detail of one of the tests performed on the UVC device. The objective of this test was to demonstrate that the standard cycle of the chronos can achieve a 4-log reduction on Mycobacterium terrae. The method used was based upon the EN standard 14563. During this test, silicon carrier were contaminated with the microbial suspension in the presence of interfering substances. Carrier were positioned in the chronos unit using a specific angle. A standard cycle delivering a minimum UVC dose was initiated, and at the end of the cycle, the treated carrier was sampled and the number of viable bacteria remaining on the carrier was determined. As you can see, the result of this study clearly demonstrates that the standard cycle of the chronos unit reached a 4-log or is able to achieve a 4-log reduction on Mycobacterium terrae spread on silicon carrier. And clearly, another time, we demonstrate that this UVC disinfection process is able to reach the same efficacy level than what is required for a chemical disinfectant. Apart from what we called harmonized standard, that means the standard used to demonstrate compliance with the essential requirement of the medical device regulation, some countries like UV have developed specific standards for UVC disinfection process. Even if this standard was developed very recently and has no direct impact on the CE mark, it's appropriate to ask whether the chronos meets the requirement of this new standard or not. In fact, the BS standard 8628 is only an adaptation of phase 2 step 2 test described in the EN 14885. There is no huge difference between this test and all the tests described in the EN 14885. Of course, you may find some slight difference. For example, the test surface is slightly larger in the British standard compared to the EN standard, but the minimum log reduction are comparable. The amount of soils required are equivalent between the two tests. And in the British standard, you can claim independently the activity, whereas for the EN standard, you have one EN standard for each specific claim. But comparing the two tests, there is no real need to repeat this test, seeing that similar tests have already been performed. So, once you have demonstrated that your process presents all the required efficacy, it's interesting to focus on specific microorganisms which are of medical interest, or because they represent a real challenge. This is the case for bacterial spores. As you know, they are considered to be the most resistant microorganism, and they are used to demonstrate the efficacy of sterilization processes, or according to US regulation, to qualify a chemical as a sterile. According to the study we have published in 2022, we clearly demonstrate that UVC disinfection process inactivated spores within 90 seconds, with a 6.2 log reduction within only 84 seconds. Furthermore, this study clearly indicates that the UVC unit was much more effective than usual FDA-approved chemical high-level disinfectant product to kill spores in real-use conditions. So another time, we demonstrate that UVC disinfection process can achieve the same efficacy level and even more on bacterial spores than already clear chemical disinfectant. To finish, I would like to spend a few minutes on the paper published by Mayer in 2017, which suggests that healthcare facilities using endocavity ultrasound probes need to strongly consider disinfection methods that are effective against HPV. In this study, Mayer demonstrates that the UVC irradiation is an effective disinfectant method to inactivate human papillomaviruses, whereas some common disinfectant may be less efficient. I now give the floor to David Weber, who will present the result of the clinical study. Thank you very much.
Thank you, Dr. Lionel Panu, for your presentation. I would now like to introduce you to our second speaker, Professor David Weber.
The audience for taking time out from your day to listen to this presentation. What I will be covering is what part of the ultrasound probe should be high-level disinfected, automation versus manual disinfection, first in-use study on human papillomavirus, or HPV, evaluation of a new UV-based high-level disinfection method, mythbusters with insights, shedding a light on UV technologies in the hospital environment, and a review of 2 studies. So what part of the ultrasound probe should be high-level disinfected? Well, we have the probe, the insertion shaft, we have the probe handle, we have the probe cable, which then leads to a plug socket, obviously, to provide electricity for the probe. Here is the Spaulding classification. This has been used now for decades. It's widely accepted around the world. It looks at the risk of infection based on how a medical device is used. And we have 3 different categories. We have non-critical under the Spaulding classification. This would be that the device, including a probe, contacts only intact skin, and the risk of cross-transmission is low. Then we have semi-critical. A probe contacts non-intact skin or a mucous membrane, and the risk of cross-infection is considered medium. Finally, we have critical. A probe contacts sterile tissue or the bloodstream, and the risk of cross-infection is high. Now, for semi-critical and critical items, we would either use sterilization, or if the device cannot be sterilized, we would use high-level disinfection. Well, let's turn now and look at the probe itself. What part of the ultrasound probe is classified as semi-critical should be high-level disinfected? Again, under the Spaulding classification, the area that comes in contact with the patient's procedure site, as you can see on the left, should be high-level disinfected. And the risk of, on the left, the risk of contamination due to damaged condoms is always present. So even if the probe has a condom, because condoms may be intrinsically with holes or may fail, we would still do high-level disinfection. We do recommend the use of condoms, but that doesn't mean that you can change the disinfection method. On the right, where the probe handle is that you're touching and which joins the cables, there is a risk of contamination through contaminated hands or gloves if you touch either the mucous membrane of the patient or the probe that's inserted itself. So then areas for high-level disinfection include the probe tip, the probe shaft, the probe handle, and the section of cable that might be contacted by the gloved hands. Okay, let's turn now to the part of the probe should ideally be high-level disinfected using evidence-based research, which eliminates guesswork. So here's some of the data based on what I've already told you. 13% of covers have tears. Pre- and post-transvaginal ultrasound examinations have demonstrated this. Before disinfection, approximately 44% of transvaginal probes had presence of bacterial contamination after the probe sheath was removed. That is, in removing the probe sheath, either the hands of the provider or the outside of the probe comes into contact with the area that needs to be high-level disinfected. This is similar to another study that found that 48% of surface probes tested positive for microbial contamination, that is, bacteria, fungi, or viruses. Let's turn now to the probe handle. 80% of probe handles had residual pathogens, including multi-drug resistant organisms such as MRSA. 62% of transducer cables showed microbial contamination, particularly in the area right near where it's handled. A meta-analysis found procedures using low-level disinfection wipes, contamination remained in approximately 13% for pathogenic bacteria and 1% for pathogenic bacteria on endocavity probes. Research shows that probes contaminated despite probe covers and low-level disinfection. There are multiple studies in the literature. One study from Hong Kong showed 7.5% were positive for human papillomavirus. Studies from France, 2 different studies, showed 3.5% and 2.8% HPV positivity after probe covers and low-level disinfection. A study from Spain showed approximately 6% HPV positivity, and another study from France was approximately 15%. Well, let's turn now to automation versus manual disinfection. Best practice in ultrasound probe high-level disinfection is achieved by manual cleaning followed by an automated system. The efficacy of wipes is user-dependent. It's easy to miss spots on a probe just using wipes. In addition, if the wipe dries out, it doesn't have adequate chemical disinfection. Use of wipes there is insufficient validation and reproducibility, which has been shown. And wiping workflow is the least preferred by actual users of ultrasound probes. Turning further to automation versus manual disinfection, again, the efficacy of wipes is user dependent. The correct application of disinfectant impregnated wipes is very user dependent, which leads to inconsistent results. It is difficult to achieve and verify compliance, to have all surfaces that have been in contact with liquid disinfectant. Not only do they have to be in contact, but they have to be in contact for the required time. Test strip may be required to determine concentration, but that's not practical in most uses. The contact time is always defined for the product, but in practice, it's often not adhered to because people really can't memorize the times in their head. Microbial efficacy may not be achieved and a stopwatch may be required to determine compliance, but this is often not practical. Furthermore, in the HSE guideline for decontamination of semi-critical ultrasound probes, the wipe system was the least preferred option and stated it should only be used as an interim measure prior to implementation of an automated decontamination process. Furthermore, let's go on continue to talking about automation versus manual disinfection. Disinfectant impregnated wipes do not provide the same quality assurance as automated devices. If you ask 20 people to wipe a probe, likely all 20 would do it differently. The Robert Koch Institute in Germany stated that disinfection with manual wipes cannot be validated on site, which is a requirement for the final disinfection step of semi-critical devices such as endocavity ultrasound probes. There is no guideline or standard to advise the requirements regarding how a manual wipe step process can be documented by an individual in a reproducible manner. Has disinfectant wipe been applied effectively to all probe surfaces? Has the correct level of mechanical force been applied to all probe surfaces? In addition, has the appropriate time, been documented? In a recent study, it was found that healthcare personnel preferred using an automated ultraviolet light system, UVC, high-level disinfection, versus chlorine dioxide multi-step wipe system. According to ISO standard 17664-1, section 6.7.1.2 states, if an automated system can't be specified, a validated method of manual disinfection is possible. Ultrasound probe decontamination that is automated for both disinfection and traceability thus ensures strict compliance with the disinfection protocol. Let's turn now to time and efficiency. One colleague, a clinical research fellow in obstetrics and gynecology at Imperial College Hospital, London, study compared Germatec Cronus UVC high-level disinfection to chlorine dioxide multi-step wipe system. The study concluded 98% of healthcare professionals found the Cronus UVC high-level disinfection easy to use and the preferred system of choice. Further, Cronus is 60% faster than chlorine dioxide wipes in the time it takes to complete HLD processes, that is cycles, per patient. Therefore, Cronus saves a total of 55 minutes, 45 seconds, allowing 3 extra patients to be examined based on a 4-hour ultrasound list of 15 patients. Well, let's turn now and talk about research and test protocols. Potency tests and in-use tests are used to evaluate the efficacy of high-level disinfection devices. There are multiple types of test that can be used. The most common are a potency test that is carrier-based. Discs represent ultrasound probe surfaces and would be artificially contaminated, and then using the system would be used to demonstrate decontamination. And then there is a clinical use test. Pathogens are placed on the ultrasound probe surface, run through the device, and decontamination is assessed. Let's talk about the in-use clinical test. This is a first-in-study research on human papillomavirus, HPV, conducted in a clinical environment. The objective of the study was to determine if ultraviolet light C spectrum, UVC disinfection, is more efficient than low-level disinfection in routine clinical conditions, especially against HPV. The study compared two protocols usually found in infection prevention practice when using endocavity ultrasound probes. The simple protocol consisted of performing low-level disinfection with a chemically impregnated wipe on a probe used with the disposable cover. The more rigorous protocol consisted of performing high-level disinfection on a probe that was partially cleaned. To the right, you can see this paper by Maxime Pinchon titled Decontamination of Intravaginal Probes Infected by HPV Using UVC Decontamination System. Now, the key results of this study showed the following outcomes. All 47 probe covers were intact, no visible damage, although, as we've already seen, they can often be damaged, and positive human and HPV DNA. Simple protocol cleaned and wiped with low-level disinfectant. 8 probes were positive for human or viral DNA, 7 probes with human DNA, and 1 with high-grade HPV 51. Now, with the rigorous protocol, UVC disinfection was applied only to partially clean probes. All probes were found to be negative. The UVC HLD procedure was found to be statistically more efficient than low-level disinfection, with a p-value less than 0.05. In the upper right, you can see actually a chart that demonstrates these numbers. Again, human DNA, chemical disinfection, positive 7 out of 47, approximately 15%. Ultraviolet disinfection is 0 over 47, 0%. HPV DNA found in 1 over 47, 2.1%, with chemical disinfection found in 0 over 47, with ultraviolet disinfection 0%. This research confirmed that UVC high-level disinfection is more efficient for ultrasound probe disinfection than low-level disinfection in a clinical use test, highlighting that UVC high-level disinfection is an easily adaptable solution to a clinical healthcare environment. Well, let's now talk about evaluation of a new UVC-based high-level disinfection method. HLD has traditionally been performed using chemical products such as glutaraldehyde, peracetic acid, hydrogen peroxide, or orthothioaldehyde, all of which have drawbacks. Recently, automated systems based on UVC irradiation have been approved in several countries for HLD on non-lumen devices such as vaginal, rectal, and surface ultrasound probes. In this study, we evaluated one UVC device, the Hypernova Cronus system, Germatech, using U.S. Food and Drug Administration, FDA, requirements criteria for liquid chemical sterilants, which are also high-level disinfectants for assessing potency carrier tests and simulated use tests. So evaluation of a new UVC-based high-level disinfection method revealed the following. We'll start with potency tests. Evaluated HLD efficacy, that is log 10 reduction, using the Hypernova Cronus system, using its standard cycle, and achieved the following results against selected microbes by inoculating a predefined area of silicon carriers, except for viruses where glass Petri dishes were used. Bacteria, greater than 6 log reduction. Fungus, greater than 4.7 log reduction. Viruses, greater than 4 log reduction. And mycobacteria, greater than 6 log reduction. Now let's turn to simulated use tests. This involved inoculation of 5 different ultrasound probe types, each tested 5 times with an efficacy target of greater than 6 log 10 reduction of Mycobacterium terrae in the presence of organic, that is 5% calf serum, and inorganic, that is 400 parts per million of calcium chloride calcium CO2-3 interfering substances. All potency tests met or exceeded the threshold requirements for microbial reduction of HLD. And I should note the inorganic load and the organic load would correspond to failure to clean the device after use prior to high-level disinfection. Importantly, no growth of microorganisms were observed in any of the simulated use test samples. The conclusion, based on methods adopted for a UVC disinfection unit, the data presented show that Hypernova Cronus system can be deemed to be compliant with US FDA requirements applicable to chemical high-level disinfection. Well, let's talk about mythbusters and insights, shedding a light on UVC technologies in the hospital environment. On the right, we have a recently published paper entitled Shedding a Light on UVC Technologies in the Hospital Environment. In this review of UVC technologies by Dr. Demersamin and colleagues, we would like to express several concerns with this review. First, the authors appropriately list some of the parameters that affect the effectiveness of UVC irradiation for disinfection. Although beyond the scope of their review, it should be noted that similar factors affect the effectiveness of chemical disinfectants, including concentration, time, duration, temperature, inherent microbial resistance, and the need for the chemical to have direct contact with the microbe. Second, with regard to room disinfection devices, the authors state that most studies reported a 1 to 2 log reduction in microbes on the surface. While this statement is correct, it is also misleading. Studies have demonstrated that UVC room disinfection devices can achieve greater than 3 log of reduction of multi-drug resistant vegetative bacteria placed in carriers. The reason that most studies only show a 1 to 2 log reduction is because that's the average load of microbes on environmental surfaces. And so even if you remove all the microbes, it's still generally only 1 to 2 logs. Third, as noted in the review, HLD requires a greater than 6 log inactivation of microbes. This can be achieved in a sealed closed chamber by UVC for non-lumen endocavity endoscopes, as the distance between the UV source and device is very small. Importantly, UVC energy decreases as a squared function of distance. In fact, one UVC device used for high-level disinfection of endocavity ultrasound probes has been demonstrated to achieve greater than 6 log decrease in microbes, including spore formers such as Bacillus subtilis, thus meeting the criteria for HLD. Fourth, the authors correctly note at the present, a UVC device for HLD has not been approved by the U.S. Food and Drug Administration for HLD of microbial devices. However, they failed to mention that one such device has been approved for HLD of non-lumen endocavity probes by regulatory authorities in France, Canada, and Australia. Finally, the lead author stated that the authors of the paper on UVC disinfection devices for HLD of endocavity probes have a conflict of interest. We take umbrage at this inference that such papers are biased or not scientifically accurate. We note that the authors of these publications disclose the conflicts and that the papers were peer-reviewed and accepted by the editors. Now, finally, Mythbusters in review of 2 studies. On the light, you see disinfection of transvaginal ultrasound probes in a clinical setting, comparative performance of automated and manual reprocessing methods. On the right, disinfection of transvaginal ultrasound probes by UVC, a clinical evaluation of automated and manual reprocessing methods. The study protocols between these papers were not the same, so it's not possible to directly compare the results of the data in these 2 studies. Again, you have to, if you're going to compare data between 2 studies, they have to use identical methodologies. So finally, ultraviolet germicidal radiation for high-level disinfection of ultrasound probes has been demonstrated to be highly effective in multiple different in vitro tests and in actual practice. Thank you very much for the ability to talk to you today and for listening to this important presentation.
Thank you, Dr. Lionel Panu and Professor David Weber, for your presentations. On behalf of Germitech, we would like to thank the people that participated in today's webinar. Thank you and enjoy the rest of your day.